Did it?
Hi, and welcome to our seminar series on the cerebellum. Today we have as our guest Phyllis Faust, who is a professor of pathology and cell biology at Columbia University. She received her MD PhD from Washington University in St. Louis. After completing a pathology residency and neuropathology training at Columbia University, she did postdoctoral training with Mary Hatton at Rockefeller, where she developed the first mouse knockout model for Zellweger syndrome. It's a condition that produces reduced CNS myelin, which fostered her interest in cerebellar pathology and neuronal degeneration. In 2006, she began working on essential tremor pathology in human autopsy brains in collaboration with Dr. Ethan Lewis and the New York Brain Bank at Columbia University. Her objective is to teach us today about how neurons undergo changes in their structure, in their axons and dendrites when they are stressed, particularly in the cerebellum. She studies brains of people who were diagnosed with essential tremor, spinocerebellar ataxia, and will describe the changes that have occurred in their cerebellum. Thank you so much for being with us, Phyllis.
All right. Thank you so much for the invitation and opportunity to speak with such an esteemed group of researchers. So as Riza mentioned, I'll be telling you about our adventures journey through the cerebellum in a spectrum of human cerebellar degenerative diseases, ranging from tremor to ataxia. Whoops. Okay, I click on it. Okay. So a wide variety of morphologic changes have been identified in cerebellum of patients with ET, as well as genetic and sporadic causes of ataxia. For those of you that may not know much about ET, it is just briefly, it's a very common chronic progressive disorder characterized by action tremor, predominantly the arms, but tremor may occur in other regions. And along with other motor features, clinically suggested involvement of the cerebellum. And you may think, well, why am I making such a big deal out of that? Because there was a time where people thought there was no pathology in essential tremor. So the changes that we will focus on today involve, hold on one second, I gotta make you all smaller. There you go, so I can see my screen, thanks. Involve the Purkinje cell, soma, its dendrites, and axon pathology, as well as interactions with basket cells and the climbing fiber afferent input. But how these cerebellar pathologies may potentially interact to delineate disease-specific changes had not been systematically evaluated. So here's an outline of what I'll be talking about today. I'll talk to you about our journey to elucidate the underlying neuropathology that does exist in the ET postmortem cerebellum, and then to look at how we can place those pathologies within a broader context of tremor and other cerebellar neurodegenerative disorders. And this is all largely published data. And the last two sections will be unpublished studies showing our characterization of ET pathological changes, not only in the motor region where we have been focusing our analyses, but also to now venture into other non-motor regions. And the last section will be a glimpse at our proteomic analysis that we've done in ET across the tremor circuit. So this really journey began in 2003 for Olan Lewis when he was at Columbia, and he established a central tremor centralized brain repository along with the director of the New York Brain Bank at that time, Dr. John Paul von Sodel. And it was really designed to address an antiquated dogma. There wasn't any pathology in ET. But that was really based on very few postmortems and poorly constructed, non-controlled, case-controlled studies. So we received brains from all across the United States. These individuals are prospectively followed by Olan Lewis to obtain a lot of clinical data as well. This is a younger me, as well as Shenghan Kuo, who eventually joined us in this journey as well. And we received brains fresh and then can obtain frozen as well as fixed tissue specimens for analyses. And we have since examined over 250 ET brains. So what are some of these changes that we have observed? So one of the first things that was identified in the ET cerebellum was that there were these increased numbers of focal Purkinje cell axonal changes. They get this cutesy name called torpedoes, which they represent a proximal swelling on the Purkinje cell axon. Shown here is this glassy pink thing. And we can use a variety of different type of tissue sections to identify these. And here shown in a Calbindin stained thick tissue section. Here you can, so we can follow the course of those Purkinje cell axons in greater detail as they traverse the granule cell layer. And you can see here is an axonal torpedo. Well, we can also identify thickened axons as well, and I'll show you additional morphologies as well soon. There are a variety of Purkinje cell dendritic changes that we have characterized. The simplest thing to observe is an increase in number of swellings of the on the dendritic arbor of the Purkinje cells. And here, this instance, you see it connected to the Purkinje cell. They can be quite large. They have a variety of different morphologies. And these are increased in the ET cerebellar cortex. We also did a large, whoops, that happens, Golgi staining study in which we got students, God bless them, to trace out all these arbors in Neurolucida software. And we quantified the changes, and we identified changes that indicated that the dendrite complexity was reduced in a central tremor, and as well as a loss of dendritic spines as well. So you can see here segments that lack any spiny branchlets coming off of it in that case of ET. We next then also identified that there are changes in this interaction between Purkinje cells and basket cells that you can observe in these cerebella. And we came up with a rating scale. But if you look using this Bilshowski silver stain, you can see in comparison to a control, which the basket plexus appears rather slender, you get an increased number and coarse thickened processes of these basket cell plexus that normally surrounds the Purkinje cell soma and inhibits them. And we believe this is a basket plexus remodeling in response to Purkinje cell loss. And we developed a rating scale centered around this that did distinguish ET versus controls. And I'll have a little bit more to say about that later. Also, there are an increased number of heterotopic Purkinje cells where the Purkinje cell soma is translocated up into the cerebellar molecular layer. You see this Purkinje cell here is sort of escaping its basket cell inhibitory input, being in this abnormal heterotopic location. And you also will often see combinations of changes here where the heterotopic Purkinje cell also has two axon torpedoes on its axon. There are also a complex set of other types of axonal remodeling and connectivity that we can observe in these thick 100-micron thick tissue sections stained with Calbindin. And we quantified these. So in addition to thickened axonal profiles and torpedoes increase, there's also an increase in the number of recurrent collaterals, and the axons tend to be excessively branched. And we can also observe incidence of terminal axonal sprouting. And so our thoughts about this is, this would alter the connectivity within the cerebellar cortex by creating new and possibly aberrant synapses. Work spearheaded by Shenghang Kuo was the fact that there is also a reorganization of the climbing fiber Purkinje cell input. So one assay is where we quantify the percentage of climbing fibers that make it into this outer 20% of the molecular layer. And that's a bit unique in that your spiny this very smaller dendrites are pretty much there. So it's really more predominantly a parallel fiber domain anatomically. And so of course there's competition electrically to synapse on various portions of the Purkinje cell arbor. And so this is reflecting some degree of invasion that we can assay of that parallel fiber domain. And you can see that that's increased in ET versus controls. In addition, we can quantify the synaptic density of these climbing fiber puncta labeled by the BGLU2 antibody. And that is reduced in essential tremor. In addition, we have identified that there is a degree of selective Purkinje cell death in essential tremor, which ranges from about at max 15 to 20% on average in the ET cerebellum by the time postmortem. And we can use simple Luxol fast blue H&E stains as well as calbindin. And you also observed that the Purkinje cell count will negatively correlate with torpedo count. So as you lose Purkinje cells, you get more torpedoes. Suggesting this may be part of a concerted process that occurs during cerebellar degeneration. In addition, we developed a second assay to look at Purkinje cell loss, and it's sort of an indirect measure of Purkinje cell loss, and it's a dual immunostain on paraffin section. So we use calbindin to identify the Purkinje cell soma, and sometimes it could be a small fragment of that. And we use GAD, glutamic acid decarboxylase, to label the basket cell processes that normally surround those Purkinje cells. And as you look, you will find basket cell plexuses where you no longer see that Purkinje cell soma. And so we call those empty baskets. And so and they are increased. And it reflects that apparently that the basket cell plexus is relatively more resistant to degeneration than are the Purkinje cell somas themselves. And this is, I think, emphasized perhaps best by this picture here in an SEA6 case. Perhaps there's a Purkinje cell here, but you see this very complex, very thick and redundant basket cell plexuses that are just sitting there in the SEA6 cerebellum at the time of postmortem. Why they're there and why they don't degenerate further, I don't know, but this is what we observe. It's an interesting phenomenon to me. So this is sort of an array of the kind of changes. So what are our thoughts on how might this lead to tremor? You guys are physiologists, so you might like blow me out of the water here, but anyway, it's just some thoughts. One of them first centering on this concept of reduced GABAergic output from the cerebellar cortex. So this is highly schematized because we know that basket cells can do innervate multiple Purkinje cells, but for simplicity, when you have Purkinje cell loss, then the surviving basket cells appear to sprout and then innervate adjacent Purkinje cells, increasing the degree of inhibitory drive to those Purkinje cells. In addition, there are these increased recurrent collaterals. So there's increase in inhibition of other Purkinje cells to their neighbors. And so in some, these will reduce the inhibitory Purkinje cell output from the cerebellar cortex to the cerebellar nuclei. And there is data from the experimental literature to support the concept that these kind of morphologic changes may increase Purkinje cell coordinated firing, so that the recurrent collaterals will do that. And these abnormal basket cell Purkinje cell interactions, both by molecular layer interneurons, as well as the specialized portion of the basket cell plexus, it's called the cerebellar pinceau. That coupling there can also enhance Purkinje cell firing, which may lead to tremor. Secondarily, of course, you all know that there's synchronous firing induced by climbing fibers. And Ming Kai and Shenghang Po published a study many years ago, starting with these findings in cerebellar autopsy tissues, along with studies in mouse models, and demonstrated this synaptic pruning deficits of climbing fibers to Purkinje cells might be responsible for tremor generation. So next we pose the question, how do we put these pathologies within the spectrum of what might happen in other cerebellar degenerative disorders? So with all these different metrics, I torture people in my lab, count this, count that. It's a lot of work, but you come out with strong numbers, and you can do any kinds of statistical and systems biology kind of methods to then look at that data. So we came into this postulated that maybe are there distinctive disease profiles that would distinguish ET from the SCAs, Friedreich's, multiple system atrophy, as well as some diseases in which the cerebellum may be secondarily affected, such as Parkinson's disease and dystonia. And so we had 37 morphologic metrics, and we grouped them into these 8 broad categories. And we published this data in a series of 2 publications. I agreed to do it once and said, why did I agree to do it again? But I did. And we ended up with a publication with a total of 320 brains. And I can only thank really the number of people that gave us these ataxia brain specimens, which are quite rare and precious. So this is just to show you again these broad categories that we studied and point out a few other things. Here's the Purkinje cell loss and this fun Kelbind-Gad immunostain. As far as Purkinje cell heterotopias, how do those come about and what does it mean? We're not really sure, but you'll see in these thick tissue sections that see how the Purkinje cell is like sideways, and here it's actually upside down, that that its dendrite arbor is facing down. So our thoughts are that with dendrite regression within the molecular layer, that perhaps the cell soma gets pulled up into the overlying molecular layer. Purkinje cell dendrite swellings is a category, these axonal changes. And note that in these thick tissue sections, we can also identify axons that may have multiple axonal swellings along the course of its trajectory. And here you're seeing the characteristic recurrent axon as well. Other outside of torpedoes, we quantified the number of axons that were recurrent, thickened, or branched, and whether it contributed to the axonal recurrent plexus that exists around Purkinje cells. And we came up with a rating for this terminal axonal sprouting as well. We observed this odd change in spinocerebellar ataxias. I think actually this probably reflects death of the Purkinje cells. They are aggregates of Kelbind cytoplasm, sort of where the ghost of the Purkinje cell used to be. And it's pretty distinctive to SCAs. We also defined in prior studies that if a Purkinje cell axon had a torpedo, it was also more likely to bear a recurrent axon or have it thickened or be branched as well. And then the last 2 categories are this basket cell axonal hypertrophy and the climbing fiber synaptic changes. So we combine all this data. We've got 10,000s of data points, and we first did principal component analysis. And if you look at this entire spectrum, it's a large number of samples with varying degrees of severity. And at this first very low power view, you see that principal component 1 largely segregated MSA here in this magenta, and SCA 1 too many is what we call them. I'll just call them SCA many after all this. And they were further distinguished by metrics in principal component 2. So if we look at correlations with principal component 2, what might be some of these metrics that are driving this distinction between SCAs and MSAs? And so anti-correlated metrics are going to bring you down in PC2 space. And those are many of these Kelbind and Purkinje cell axonal changes, whether it's thickened or recurrent, and these torpedo-associated changes, and numerically the number of torpedoes as well. And overall somewhat lesser variable degree of loss in MSA, whereas in SCAs, you have a lot more of these dendrite swellings and a greater degree of Purkinje cell loss is emphasized by more empty baskets. If I'll just show this briefly, if you look at a principal component analysis to segregate these different types of SCA cases, we had also an SCA 8 and 14, but not enough to graph on this. You see a spectrum of changes with SCA 3 and Friedreich's ataxia being on the mild end, and SCA 6 showing the most severe changes, which is consistent with the fact that the cerebellar cortex is a primary site for degeneration in that disorder. So here's just some histologic pictures. Pictures speak 1,000 words and numbers. So just to show you the kind of spectrum of changes that you might see. And so here in this SCA1, for instance, you see a lot of Purkinje cells are bearing axonal torpedoes. There is a reduced density of axons in the white matter, reflecting the fact that many of them don't make it down there, but instead probably become recurrent and inhibit their neighbors. And we're seeing that it's a spectrum. And by the time you get to a severe cerebellar degeneration, you have very few Purkinje cells, but most of them do bear axonal torpedoes. In MSA, I'm going to talk a little bit more about this. We see a wide spectrum that does correlate with clinical phenotype. And I really love this picture. It's one of my favorites, but just so many torpedoes that you have in these MSA brains, even more sometimes in these SCA brains. And many of them are having these recurrent axons and not making it down into that white matter. So if we remove that MSA, the severe degenerations, we can break apart that very tight cluster. And we do see that there's a lot of overlap. The concept of what's going on in Parkinson's is a topic for another study. But the ET cases are more shifted to the right than the controls are. And all the kind of metrics that drive this PC1 segregation are exactly the kind of things that we just talked about. The torpedoes, dendrite swellings, and the climbing fiber changes, etc. And so we've postulated in the literature that ET is a mild form of a cerebellar degeneration, and that it can be segregated, as shown here, from many of the more severe degenerations as far as its severity, with these array of metrics. So let's look in a little bit more detail how these differences in fold change versus for each metrics across these disease states, grouped into these 8 broad categories. So, and we set a fairly tight significance level for these multiple comparisons. So for dystonia and Parkinson's disease, we only saw a spattering of changes across these different metrics, whereas in essential tremor, there was a lot of changes. A fewer numerically number of changes in Friedreich's ataxia and SCA3. And many have postulated that the cerebellar cortex is normal in those individuals, but certainly by the time of death, there are degenerative changes that occur in the cerebellar cortex. And MSA and these SCA many kind of cases, there's a lot of changes. In addition to a lot of changes, they are also numerically greater, okay? I'm not showing you, but we also did some hierarchical clustering of correlation coefficients between all pairwise combinations of metrics. And I'm mapping it back to here, and we identified about 25 of these metrics that had highly correlated changes, suggesting they're part of a repertoire of entities that change during cerebellar degenerations, and mapping them back here. And we call these core signatures of cerebellar degeneration that we have identified. More pictures, because they're fun. So there are commonalities between ET and cerebellar degenerations with similar Purkinje cell axonal pathologies. They are numerically more prominent in essential tremor than they are in Friedreich's ataxia, but their morphologies and alterations are similar. Again, ET in concert with SEAs, you'll often see these clusters of these axonal torpedo-bearing axons, with many of which have recurrent collaterals. That's a phenotype that you'll see here, even in the SEAs. But in addition, we observed these similar highly degenerate Purkinje cells. And people have described these previously in rare publications in SEAs. And once you see them, you start seeing them sort of all over the place, in which you get this sort of halo-like arrangement of multiple thin dendrites emanating from the Purkinje soma and some somatic spines. And sometimes you'll see residual dendrite swellings associated with these cells. And we think that perhaps this is like a regressive kind of phenotype, perhaps honing back to a time when Purkinje cells did not have such polarized dendritic arbors. And so, of course, regressive changes can be part of a degenerative process as well. And in addition, these dendrite swellings, was not actually part of this study, but we just recently published this. And they may have a variety of different morphologies, some of which are quite interesting. So shown here, you can see an array of thin spiny processes emanating from them, shown here in the Kalbinden stain. And particularly if the dendrite swelling is larger in size, it tends to have quite complex morphologies as shown across these different disease entities here. And we also identified Purkinje cells in which both harbored a dendrite swelling as well as a torpedo, but numerically torpedoes are about tenfold more common than are these dendrite swellings that we defined in this study. So morphologically, another thing that we observed is that there was marked thinning of this portion of that dendrite between the main Purkinje cell dendrite trunk and the swelling, and that distal to that, you would get a lot of degeneration of that Purkinje cell dendrite. It lacks any spiny processes emanating from it. And you can also sometimes observe multiple dendrite swellings on a single Purkinje cell. So obviously this is also going to cause a lot of aberrations in the way that these cells function.
Sorry, I have to cut you off just real quick.
Hello? Okay, yeah. So what's in these things? So we did do a study looking at the morphology of these axonal torpedoes, and as you would expect, they do
Phyllis, we
reflect.
lost your voice.
Oh, hello? Can you hear me now?
Yes,
Okay, where did you lose me? How many slides ago?
just 10 seconds ago.
Okay, cool. Thank you for letting me know. So the axonal torpedoes do contain a whole lot of disorganized neurofilaments and scattered degenerate organelles, reflecting that it's a disruption of axonal transport. We did not look at Purkinje cell dendrite swellings, and I only found a few rare studies in which they did, which all share the fact that they were performed in the prior century. And in contrast, dendrites intrinsically have a larger content of organelles than do axons. And so in the diseases where they looked at it, you do see predominant accumulation of organelles in these dendrite swellings. But how exactly these are going to disrupt cellular function, and in particular for axonal torpedoes, are they purely degenerative, or do they also represent a reparative, regenerative kind of response for the cerebellar Purkinje cell? So further looking at patterns of changes across diagnosis. So we computed some scores, we combined fold changes for selected metrics. And so if you look at us, we combined 25 of these positively correlated metrics, and this is the spectrum of kind of changes. As you go from left to right, you see the same kind of change, but just more of it. So that is a predominant pattern that we observed in this study. Purkinje cell loss, as we've been saying, is more prominent in essential tremor, but it's widely variant in multiple system atrophy and far more severe in the spinocerebellar ataxias. But the pattern for the climbing fiber metrics is interesting and perhaps revealing. So essential tremor has more of these climbing fibers in the outer 20% molecular layer, as I discussed. We also we only had a small cohort of Friedreich's ataxia cases to examine in this assay, but so ET is the only, excuse me, disorder in which you have reduced climbing fiber synaptic density, but invasion of this parallel fiber domain. In Friedreich's ataxia, you don't have climbing fiber overgrowth into that, synaptic density reduction, but you do have some overgrowth. Whereas in the cerebellar ataxias, they're really characterized by marked loss of climbing fiber input. So this is a paper, we started this kind of a comparative analysis between ataxias and essential tremor and controls a long time ago, and published a study in 2017. And, spearheading off of this is the concept to use animal models, and that's what Shenghan and Ming-Kai have done. And I'm just showing you the human data because they've done a lot of studies, and hopefully this is about to be published soon, demonstrating that these climbing fibers may result in loss of cerebellar rhythm and contribute to ataxia symptoms as well. And so these are the assays I've been showing you, but they also actually constructed this differently, because the fact is that they constructed a 50% line, because many of the climbing fibers don't even make it out that far in ataxias, at least at this end stage. But in animal models, obviously, you can follow this more directly, and they've done a lot of interesting stuff with that. So be on the lookout. So I wanted to talk a little bit more about this variable type of pathologies that you observe in multiple system atrophy, because I'm not sure, but it may be indicating a sort of pattern by which Purkinje cells react during cerebellar degeneration, because these patients obviously have a variety of different clinical phenotypes. So an individual that pretty much only had striatonegral degeneration, the changes that we observe in the cerebellar cortex are milder. And many of these individuals, for a subset of these MSA cases that we looked at, we knew what the initial clinical diagnosis was for these individuals. And so this is someone might be just had Parkinsonism, okay? Not much is going on in the cerebellum, but thing about multiple system atrophy, it progresses and may, may then invade into cerebellar region. In someone with prominent ataxic syndromes, as their initial presentation with MSA, those are these patients here up in red that are intermixed with the SEAs. So they're no longer, by these metrics that we have here, clinically distinguishable from an SCA, at least with this. But this stage here is interesting to me, because this is the stage when you get these numerous axonal torpedoes and all these recurrent collaterals. And so is there as the cerebellum gets involved, is this a stage of progression that occurs? Whether this might occur also in some of the SEAs, I don't know, or it possibly could be unique to MSA, and effect of synuclein oligodendroglial opathy on myelin integrity and axonal stability. And that remains to be investigated. And so another thing that sort of supports this concept is we did a prior smaller study looking at the relationship of Purkinje cell loss and torpedoes in cerebellar disease. And if you mapped out this data in this much smaller data set, we saw this sort of curve fit more of a quadratic kind of thing. And this is our current data set, and we've really recapitulated that finding. So these are these MSA cases. So for a particular level of Purkinje cell loss, you see many more torpedoes in these MSA cases, giving this positive aspect to the curve. But eventually, and so either all the Purkinje cells have torpedoes at this stage, and or multiple torpedoes potentially, but eventually the loss of Purkinje cell catches up to it, and you end up with a paradoxical sort of loss of these observable axonal torpedoes, perhaps then reflecting the degenerative aspect of this molecular morphologic change, along with other factors. So conclusions for this part of the study is that, a big finding in here was that these metrics really do, across cerebellar disease, reflect differences in degree rather than kind of metrics. Same kind of changes occur, sort of a stereotypic cellular response that the cerebellum undergoes. But they don't form a homogeneous entity. And so particularly with the climbing fiber metrics, we're seeing unique patterns of climbing fiber changes. And we believe that this data supports the concept that there is cerebellar degeneration in ET on a milder end of the spectrum, and that there's a somewhat distinctive signature that you can tease apart in each of these cerebellar disorders. So I want to go on next week. All of our studies to date have really focused on the anterior motor cortex. Seems obvious why you'd want to look there in motor disorders. And we had yet to address whether, okay, are there changes elsewhere in the cerebellar cortex? particularly in these non-motor regions I've outlined here for lobules Crus 1 and Crus 2. So by doing that prior study, we've now sort of pared down our metrics to 11, which is a lot nicer, huh? This was led by a very talented research staff associate of mine. And this is, now we're down to 11. That's nice, huh? The same kind of metrics that I've just been talking to you about that we're continuing on with. So a couple important messages that have come out about this study. So first of all, in controls, Purkinje cell density, linear density is lower in the non-motor Crus 1, Crus 2 region than it is in the anterior motor cortex. And we're surprised by this initially, but then you read the literature, and this has been previously reported in a small stereology study and also in some animal model studies as well, that there's an inherent difference in the packing density of Purkinje cells in this non-motor region versus motor region. You see the same kind of pattern in the ET cases, but nonetheless, you can still distinguish between control and ET by a reduced Purkinje cell density in both regions, okay? So you detect it in both regions, but there does appear to be a somewhat greater loss of Purkinje cells in the motor region. So the important thing about this is these 2 points here. So, and that you carefully sample where you are in the cerebellum in any sort of study. And it's not always specified, believe it or not, in many of these human studies where they're taking their cerebellar sample from. And it's important because you can see that this mean is approaching that one. And so it could lead to a failure to detect a case control difference if you don't carefully sample from the same regions. How about those climbing fiber changes? So in contrast to Purkinje cell density, for controls, we're not seeing differences in the climbing fiber synaptic outer 20%, as well as the climbing fiber synaptic density. But we do still observe disease-related changes in ET, both in the motor and the non-motor region for both the outer 20% and the climbing fiber synaptic density. Although again, the burden is higher in the non-motor region. So this suggests that there is more widespread cerebellar involvement in ET, although with greater disease burden in the motor cortex over that in non-motor cortex. For some other metrics, again, we see similar pattern for torpedoes and dendrite swellings, where it's ET, you can distinguish ETs and controls in both regions. Sometimes the torpedoes are again not differing, or dendrite swellings in controls, but they do distinguish ETs between these regions and ET versus control in both regions. And an unexpected finding was that heterotopic Purkinje cells are much more common in the non-motor region than they are in the motor cortex. And similarly so in both controls and ETs, about 4-fold higher in the non-motor region versus the non-motor region. What this means and why, I do not really know. I only found one study done in a dystonia mouse model where they observed a similar increase in ventral regions of cerebellar cortex for heterotopic Purkinje cells. Now, again, if you combine these metrics, we can observe different patterns, and we are seeing some distinctive clusters that come out of this kind of principal component analysis that can define these different regions in these comparisons with significant differences across these principal components. So this is a way that we can identify patterns. And so in some summary for this part, pathological changes are also present in the non-motor region. They similarly distinguish ETs and controls and demonstrate more widespread involvement of the cerebellar cortex. Now, do they contribute to cognitive changes that do occur with increased incidence in ET patients? If we just take simple metrics, is the patient demented versus not, or has mild cognitive impairment, we're not seeing good correlations, but we do have very good clinical cognition data on these individuals, and with more refined metrics, I think that there are some changes, and we're working on that right now. Again, the burden of pathological changes is greater in the ET motor region than non-motor region, consistent with the prominent motor symptoms in this disorder. But perhaps a most common pattern of cerebellar degeneration is that of an anterior posterior gradient, which currently this data seems to indicate that this degenerative process is following. But we are continuing our studies to look at other cerebellar regions as well to see if this pattern holds up. But the take-home message also is it's very important to sample equivalent cerebellar regions when studying these degenerations. You would think that'd be an obvious thing, but it's really not always true in studies that I've read. Now, the last section that I wanted to show you is some of our more molecular studies. And this is some preliminary data. So we've done some proteomic studies using these frozen tissue samples and looking at regions across the tremor circuit involving the cerebellar cortex. And we also did laser capture microdissection to isolate the Purkinje cells plus molecular layer to hopefully enrich for Purkinje cell related transcripts. And we also lasered out the dentate and the inferior olive, and then did proteomics on them. And these are studies spearheaded by Dr. Marticello, who was an associate research scientist in my lab, and now being led by Dr. Oliveros Giovanni, who's a very talented postdoctoral scientist. And so we're seeing a large number of protein differences across these different regions, particularly in the whole cerebellar cortex and dentate. And many of these are quite avid changes with at least greater than twofold changes. Lesser degree of changes in the isolated cerebellar cortex molecular layer or the inferior olive. Now, obviously, I don't have time to go through this entire large data set, but at least as far as the whole cerebellar cortex, the top overlapped KEGG pathways, centered around things that you would expect, or calcium signaling, proteasome assembly and functioning, disruptions of axonal transport, and regulation of microtubule growth, which are pathways that we have described in previous RNA sequencing experiments in essential tremor versus control cerebellar cortex. But if we looked at proteins in pathways, we've curated this in multiple different ways that were expressed in at least across two regions. We've done some initial studies looking at neurofacin because that showed changes across all 4 regions of this tremor circuit. So neurofacin is a immunoglobulin cell adhesion molecules, multiple roles in the nervous system. And there are two main isoforms, with neurofacin 186 expressed by neurons, whereas neurofacin 155 is expressed by oligodendrocytes. And there are actually isoform-specific antibodies available, and they do work really well on tissue sections, which has allowed us to parse out potentially these differences. So for neurofacin 55, and I'm showing you the just data in ET cerebellar cortex and looking at granular layer axons, we've identified that there appears to be a differential localization of this protein in controls versus ETs. And in a thin paraffin section, what you observe in a control is you do see a concentration around the nodal region, but then a gradient of expression that sort of extends from that nodal region, okay, shown like here. Whereas in ET, we're observing much more localized concentration at the nodal region and a reduction in that gradient outside of the nodal region. Now, in a thin paraffin section, it's hard to sort of do percentage of co-localization. So we did a thick tissue section analysis with an analyzed co-localization with Inamaris software and identified in the white areas are regions where there's overlap between myelin basic protein and the neurofacin signal. So in the controls, you see a lot more of these solid white regions versus in ET, we're seeing more concentrated paranodal distributions. And this is reflected in the quantification here, even with a relatively small number of cases, that we're having reduced overlap in the ET cortex versus controls, suggesting maybe there's some sort of oligodendroglial dysfunction going on here as well. And so this is not specific to Purkinje cells. So we have done some studies looking at myelination patterns on axonal torpedoes. So here's a pretty cool picture. So you can see the beginning of myelination after the axon initial segment in these normal appearing axons. And this torpedo, they don't have any myelin, both around the torpedo or in segments distally to that torpedo. And here you see absolutely no myelin along this segment. However, we have observed some axons that do have myelin distal to it. And here's one such example. And if you zoom up on that a little bit, what you observe is that I believe that this torpedo, which is right here, is in a paranodal location. So this is a break in the myelination, which is probably a widened internode, which is abnormal unto itself. Then we have a small segment of myelin that exists, and then we get the torpedo. So again, this is suggesting perhaps maybe there is some nodal dysfunction in Purkinje cell axons that's contributing to these formation of swellings, and potentially leave you with this food for thought that maybe this also contributes to Purkinje cell degeneration in these cerebellar disorders. And it's not only our studies, but like there are transcriptome studies in essential tremor as well as in spinal cerebellar ataxias that are also indicating a role for oligodendrocytes in neuronal degenerations. And perhaps that's what's going on a lot in multiple system atrophy also. So that's where I'm going to finish. And I thank you for your attention. And I also thank the many people in my lab that have counted these myriad of things, as well as the families and patients that participated. And I'll take any questions. Thanks for your attention.
Thank you so much, Phyllis. Please just go ahead and raise your hand and I will call on you if you have a question for Phyllis. Phyllis, maybe I can start by asking you about the relationship between the recurrent collaterals for the Purkinje cells that seem to increase in ET and those deficits that you showed in the axons. Is it that the Purkinje cells in ET are less likely to reach the nucleus?
To some extent, yes. We haven't. We're actually sort of in the process of trying to look at the terminals in the cerebellar nuclei. You know, we have looked at the cerebellar nuclei in ET. We don't see a major loss of cell populations. In the SEAs, you do see different patterns of degeneration in the Purkinje in the cerebellar nuclei, but nonetheless, yeah, they would affect that. Those axons are not going to make it down to the cerebellar nuclei, as well as the effect that they're going to inhibit their neighboring Purkinje cells, and that would change the physiology within the cerebellar cortex. That's our thinking on it.
Sasha, you have your hand up.
Yeah, sorry, I don't have a camera working on my thing, but Phyllis, first, that was just really inspiring. Like I find it amazing that you're able to do proteomics and then to do the immuno in humans that looks good enough that you can actually make conclusions. And so the whole thing is very cool.
Thank you.
Like, way to go, like so cool. And yeah, everybody on the talk, you all know this, but really tell all of your colleagues who don't know what we know about the cerebellum that where you look really does matter because it's a huge effect, right? So I have a few questions. And the first one is this interesting observation of the basket cell like ghosts and how stereotyped that is. And it just reminded me of Jack McMahon's findings decades ago, like when I was a graduate student. He was looking at the development of the neuromuscular junction. And the question is, do the neurons cause it, or does the muscle cause it, or what anatomically, what's the deal? And so he worked out killing in his lab both, and doesn't matter. You don't need a neuron and you don't need a muscle. What you still get, all of the synapses get formed in the right places if you don't have either. What you need is the extracellular matrix. And so he identified this molecule called aggrecan as an important factor. And so just like, it just kind of makes me think, you know, it's going to be some sort of extracellular matrix, perineuronal net that doesn't get degraded by activity versus the nets around. So that's the comment more than question. Like, have you thought about that, I guess?
Yeah, I've thought a lot about them. But like, as you say, the tools that we have available to look at them are challenging in the human autopsy brains. But yeah, I think that that's potentially a really interesting avenue to go down.
Yeah,
Thank you
cool.
for that
So
comment.
you're welcome. Yeah. So the second question is kind of about like how much of this was established early in development that something was screwed up, and then there was a hit in the adult that caused this regressive, this sort of changes, and how much of it is just a complete remodeling of synapses that occurs for some other reason. I don't know what that would be though.
You mean like in essential tremor itself?
Yeah,
Or
yeah, for example. Yeah, just trying to understand sort of like, is there a developmental aspect to this that is sort of occult, or is this really an adult thing?
unfortunately, we can't, like the youngest individual we have in our brain bank from ET was like 64. Okay.
Right. Yeah.
We can consider them a young adult. In our study, they relatively
Thank you. Perfect. I love that.
but yeah, so like we don't have samples to address that. So it's really going to take different kind of modality studies to really address that question. These heterotopic Purkinje cells, we've touted that they are neurodegenerative, but we've haven't published this yet, but we looked at a large cohort of younger individual controls, okay? And they also have a lot of these heterotopic Purkinje cells. So there may well be a developmental aspect to that, although you still see far more in ET, even at matched ages. So I think it may be a combination thereof. But we just don't see this extent of changes in a individual that's called a control at the time of death.
Got it.
So
Yeah.
they are disease associated changes in our mind. Okay.
Thank you.
But when exactly they occur, it's hard to know because individuals with ET, they may start with symptoms as early as childhood. Okay. So what's going on? But
Oh,
yeah.
I didn't know that.
Yeah,
Okay.
so there's different patterns of cerebellar loss in ET patients, but those that present with disease later in life do tend to have more degenerative pathology than those with longer durations.
And a final kind of wacky question, slightly wacky. What do you like? If you could like feed, just go forward like another several decades, where will this lead in terms of diagnosis, treatment? What's your like best kind of hope guesses?
Well, there's stuff I didn't get to present today. We have identified defects in ryanodine receptor signaling, RYR1 in essential tremor, and there are like small molecule compounds that can fix that defect. Okay, so it may be sort of a common pathway, but which feeds into calcium signaling inside the cytoplasmic calcium signaling in Purkinje cell neurons. So there may be targets for that. There are, Alon is involved in some studies where they're trying to use PET imaging to look at ET patients while they're alive, obviously, to try to diagnostically distinguish them better. Okay. But the main issue is really we still are limited in the number of pharmaceuticals that we do have to treat this entity. Okay. But hopefully maybe by looking at, this proteomics data is a large data set, and hopefully it's full of rich kind of information that may hopefully lead to further insights as well. But again, it's all end stage disease, but that's what we have to work with.
Very cool. Thank you.
Thank you, Sasha. Thank you, Phyllis. Phyllis, we have a couple more questions for you. There's one question here, and there's one also question by Aditya. So I'll start with here. Alden has a question.
Yeah. So by the way, very impressive data set. It was very, very great talk. I had a question. So you mentioned, I'm not super familiar with essential tremor, but you mentioned that one of the hallmarks is an action tremor, whereas in cerebellar ataxia, we tend to see more of an intention tremor. You present a goal, and the goal, when they're presented with a goal, they're not able to make an accurate movement to it. But if you remove the goal, then they're able to make accurate movements, or their oscillations slow down. I was curious if you had seen anything in the differences between the two, between the ataxias versus essential tremor in either, the incoming mossy fibers, granule cells, or where it comes to the Purkinje cell with the parallel fibers, because we tend to think of the goal to come in through that pathway.
Mm-hmm.
And if there's what your thoughts were on the differences maybe between those two.
My thoughts is that they are good targets to look at, but that are difficult to quantify. So, because there's so many parallel fiber synapses, and it's dangerous to compare intensities in human autopsy tissues of the signal. You can do that in a mouse, okay? We have a lot more control. So like you could use vGlut1, for instance, okay, to label the parallel fiber synapses, but you just get a whoosh, okay? And it's really hard to distinguish those pathologies. So we do need a better way to look at that. Eric Carlson did do a study in Alzheimer's cerebellum, looking at mossy fiber inputs that he presented at Gordon Conference about nearly 2 years ago now. And he saw some changes there. So that is another place that we could go to. These kind of changes in neurofasten that I described to you, that's not specific to Purkinje cells. That's all axons, and probably many of those are afferent inputs. But you're right, that does need a little bit more detailed input. As far as intention tremor variability does occur even in ET as well. Okay. It's a bit of a mix. So it's not unusual to see both kinds of tremor in ET as well. But yeah, it's just been difficult to define. It's like an obvious target to like try to look at the parallel fiber synapses, but we need better methods to do it. I'm not sure.
I see. Thank you.
Thank you for your question.
Aditya, let's have the final question.
Thank you so much, Dr. Faust, for the wonderful talk. I'm curious about the heterotopic Purkinje cells that you showed, and they are more common in ET, as you mentioned. I was wondering about the difference in molecular identity of, if we know about how different they are from the regularly organized Purkinje cells in these cells, and what role would there be of this heterotopic organization, like in the disease phenotype?
Yeah, well, we don't know on a molecular level what the difference is between these cells. We know on a morphologic level that they look different, and there can be their polarity can be different relative to what it should be. Actually, I had Dr. Marticella, who left my lab. She actually did a heroic study, laser capturing out individual Purkinje cell bodies, okay, to get enough for RNA sequencing, and you just wouldn't be able to get enough of these cells nor identify them with our methods to really look on a molecular level, and we haven't seen a marker. But one thing that I do observe is it's probably they are lacking this basket cell fiber inhibitory input in large part. Okay, so that's certain physiologically one thing that would differ about them. Okay, that's about the best I can do.
Yeah, fine, thank you.
Florian, go ahead.
Thank you. Thank you. Great talk.
Thank you.
I'm saying this as a non-molecular person. Initially you had this, like very early on, you had this slide that showed a Purkinje cell kind of escaping into the molecular layer. And you talked about the pathological changes in the basket cells. And you said this might be an adaptive change due to the degeneration of the Purkinje cells. Could it be the other way around, that the basket cells create kind of a neurotoxic environment, killing off those Purkinje cells or
Yeah.
inducing changes?
That's a really interesting thought. Thank you for that feedback, because yeah, it's absolutely positive, not possible. The thing is, with human studies, anything is possible. I don't know, have there been studies where they've driven basket cells and in some sort of a slice culture or something? Does it do anything? Or so I don't know the answer to that, but you guys all have to figure this out for us. So, but yeah, that's an interesting thought. Thank you.
Wonderful. Phyllis, thank you so much for this wonderful presentation. And thank you, everybody.
Thank you for your invitation.